Number 1. Enumerate the mode of gene action and determine the phenotype and genotype of the progenies produced in each mode. There are 4 modes of gene action including additive, complete, dominant, epistatic, and incomplete dominant. Additive, both alleles equally and directionally contribute to the generation of the trait when neither is dominant, and between the two homozygous phenotype, the heterozygous phenotype is in the middle. Epistatic interaction. The generation of a phenotype is controlled by epistatic gene interaction when particular arrangement of alleles from various genes are required. Epistasis is the process by which alleles from two or more loosely interact to create a phenotype that neither gene alone would be able to create. Complete dominant. There are only two phenotypes when full dominance is the manner of gene activity, and three phenotype because in the heterozygous two different type of alleles for a single trait. Condition: the expression of the recessive alleles is hidden by presence of dominant phenotype. Incomplete dominant. When the dominant alleles manifest itself, more dominants take another from stranger than the recessive alleles but not sufficiently strong to match the homozygous dominant phenotype in the heterozygous state. We refer to this kind of dominance as incomplete dominance. Three genotype and three phenotype are produced by genes with this method of action. Number 2. Differentiate dominance from a recessive gene. Dominance refers to a relationship between two versions of a gene. If one is dominant, the other one must be not dominant. In that case, we call it recessive. A dominant gene or a dominant version of a gene is a particular variant of a gene which, for a variety of reasons, expresses itself more strongly all by itself than any other version of the gene which the person is carrying. In this case, the recessive. Now, it usually refers to inheritance pattern frequently used in conjunction with a Punnett square, where if an individual has two versions of a gene and one is observed to frequently be transferred from one generation to another, when it is called dominant biochemical. What is going on in this case is that the genetic variation, for a variety of a reason, can either induce a function in a cell which is either every disadvantageous or every detrimental, which the other version of the gene can cover up or compensate for. In the case you're going to have a dominant mutation and that dominant mutation can be benign, it can refers to a color of one sort of another that can become dominant mutation, or it can refers to a disease. Huntington disease, for instance, is dominant mutation where if one is carrying that version of the Huntington gene, that dominant mutation will give the individual the disease regardless of what that person other Huntington's disease gene alleles is. That other Huntington's disease genes alleles can be perfectly normal, but the person still has the disease because of that one copy of the Huntington's disease genes that is mutated. Number 3. A dominance G allele is common car produce normal pigmentation, and the recessive allele G alleles produce gold pigmentation, a cross between normally pigmented and good and gold common carp produce on normal progenies. Letter A. What is Letter A. What are the genotype of the parents? GG normally pigmented, GG paisonemic pigmented, GG golden. Letter B. What are the genotype of the progenies? Female GG, Gmales GG. Letter C. What genotype should be made to produce all gold phenotypes? It is the GG genotype to produce gold pigmentation. Number 4. Based on the theory discussed on the hybrid crows. Letter A. List all the gametes produced by the hybrid crows of the homozygous group guppies with curved spin. Gametes for CU. Letter B. Draw a Punnett square and determine the genotypic ratios are expected from the cross. Male gametes GZU. Female gametes GZU, GGZU, CU, GGZU. Tas Number 5. Based on the given example on X-linked genes. Letter A. How are transparent-tailed guppies produced? A transparent female guppies can produce if the female is transparent and the female has at least XEH allele on the transparent tail. Male guppies has the recessive SEH allele produced only when female. Letter B. Prove your answer to a male gametes. Female gametes, transparent tail pended transparent tail female. Letter C. Draw the Punnett square and determine the phenotype and genotypic ratio of the progenies of the cross between the mating of a heterozygous caudalous female and transparent tailed male. Caudalous female and caudalous male. Transparent tailed female. Transparent tailed male. Number 6. Determine the factor that causes variation among organisms. Mutation, the change in the sequence of genes in DNA are one source of genetic variation. Another source is gene flow or the movement of genes between different groups of organisms. Finally, genetic variation can be a result of sexual reproduction, which leads to the creation of new combination of genes. Number 7. Understand and apply the concept in selection in fish stock management. A stock assessment in the process of collecting, analyzing, and reporting demographic information to determine change in the abundance of fishery stock in response to fishing and to the extent possible predict future trend of stock abundance. The purpose of stock assessment is to provide information to fishery manager that will allow them to control the catch of other species or species complex or the effort directed to it so that ideally population can be maintained to produce the MSY or LTPY for each species. Fisheries management aim to achieve the optimal and sustainable utilization of the fishery resource for the benefits of humankind while safeguarding the ecosystem. Modern fisheries management is based on scientific information that is used to develop the rules under which the fisheries operate. Number 8. What is genetic drift and how can it be prevented? The regular replacement of foundation animal from genetically fixed embryos is the only ways that effectively slow down genetic drift and prevent genetic change from accumulating.